Wernicke encephalopathy is an acute neuropsychiatric syndrome which is caused by thiamine deficiency. Wernicke encephalopathy has three major features. It's encephalopathy, oculomotor dysfunction, and ataxia. These three symptoms combined we call Wernicke triad. With time, thiamine deficiency can become chronic, and chronic thiamine deficiency can cause Korsakoff syndrome. Korsakoff syndrome is a chronic memory disorder caused by severe chronic deficiency of thiamine. And Korsakoff syndrome also has three major features. It's memory loss, confabulation, and personality changes. So initially, thiamine deficiency can cause Wernicke syndrome, and only with time, if thiamine deficiency will last for a prolonged period of time, in addition to Wernicke, Korsakoff syndrome will develop. Together, we call them Wernicke-Korsakoff syndrome. So the first question is how thiamine deficiency develops. And to understand this, we have to know a thiamine pathway in the body. The major source of thiamine is diet. When we consume thiamine-rich food as wild grain, brown rice, nuts or bread, thiamine in comes to the small intestine. In jejunum and ileum located a specific intestinal ATPase. Exactly this transporter provides the absorption of thiamine. ATPase uptakes thiamine from GI tract and transports thiamine into the blood. And the normal level of thiamine in the blood is 50 to 200 micrograms or 2.5–7.5 nanograms. In the bloodstream, thiamine circulates through the body until some tissues will uptake thiamine to use for own needs. And the most thiamine-dependent tissues are liver, brain, and muscle tissues. Once thiamine enters into the tissues, thiamine undergoes phosphorylation, and phosphorylation is possible only in the presence of magnesium. This reaction results in production of thiamine pyrophosphate, which is activated form of thiamine. Exactly this form tissues can use as a cofactor for enzymes. In this form, thiamine can remain in the organism for approximately 12 days, and after 12 days we excrete thiamine through the kidneys into the urine. The first factor that can cause thiamine deficiency is decrease in thiamine intake, which is possible if person mostly consumes products with low amount of thiamine, as polished rice or processed grains. With decrease in thiamine intake, thiamine level in the blood will progressively decrease, and also income of thiamine to tissues will decrease, and with time this will cause clinical symptoms. The second reason of thiamine deficiency is chronic alcohol consumption. Because alcohol inhibits the function of ATPase in the jejunum and ileum. Without this transporter, we cannot absorb thiamine. So thiamine level in the blood will decrease, and thereby thiamine income to tissues will decrease. In addition to this, alcoholics often have poor nutrition. And this makes sense because why to spend money on nuts and bread if you can invest them into vodka? So, in addition to poor absorption, alcoholics also have low thiamine intake, and this only aggravates thiamine deficiency. In pregnancy, a condition known as hyperemesis gravidarum can develop. First of all, hyperemesis gravidarum can cause severe nausea. With nausea, food intake decrease, this causes decrease in thiamine level in the blood, and thereby the income of thiamine to tissues will decrease. In addition to this condition often causes vomiting, which only aggravates thiamine deficiency. Another factor that can cause thiamine deficiency is chemotherapy. One of the most common side effects of chemotherapy is injury to the small intestine. In this case, with damage to intestinal ATPase, the absorption of thiamine will decrease. Thereby, thiamine level in the blood will decrease, and with thiamine deficiency, the income of thiamine into the tissues will be low. Also, in case of gastric bypass surgery, the intestinal surface area decrease, and the lesser is the surface area, the lesser becomes the absorption. So it's another reason why timing deficiency can develop. And also, we have to remember that the essential element for timing activation is magnesium. And with deficiency of magnesium, the activation of timing decreases. So even with normal timing level in the blood, person with very low magnesium level can experience timing deficiency symptoms. So how timing deficiency affects normal metabolism? And to explain this, we have to know biochemical pathways where timing plays a crucial role. On this fancy picture, we can see a complex of biochemical reactions where timing used as a cofactor. But because it's all at once, the entire picture looks complicated. So to understand the role of timing better, we will simplify and discuss it step by step. So when glucose enters into the tissues, one of the pathways that utilize glucose called pentose phosphate pathway. And one of the crucial enzymes in pentose phosphate pathway is transketolase. The reason why transketolase is so interesting is because this enzyme uses timing as a cofactor. Simply speaking, this enzyme cannot function without timing. Pentose phosphate pathway results in production of ribulose 5-phosphate and a lot of NADPH molecules. By the way, this pathway is the major source of NADPH molecules in our organism. From ribulose 5-phosphate, we make ribose 5-phosphate, which we use for nucleotide synthesis. And NADPH molecules we use for reduction of glutathione. It's extremely important reaction for antioxidant defense. Recall that oxidative stress is characterized by excessive production of reactive oxygen species. Initially, superoxide produced, and then part of superoxide molecules are converted into hydrogen peroxide. Hydrogen peroxide is a highly dangerous substance that can cause severe oxidative damage. So to prevent oxidative damage, we have to convert hydrogen peroxide to water. And detoxification of hydrogen peroxide is provided by glutathione peroxidase. Glutathione peroxidase reduces hydrogen peroxide to water. In this process, two molecules of glutathione gives way to hydrogens. As a result, glutathione disulfide is formed, and in this form, glutathione molecules are inactive. So to become functionally active again, glutathione disulfide must be reduced to two glutathione molecules. And this conversion is provided by glutathione reductase that use NADPH as coenzyme. In this process, NADPH molecules donate 2 electrons to glutathione disulfide, thereby converting it to 2 glutathione molecules. And now these 2 glutathione molecules can be used by glutathione peroxidase for detoxification of hydrogen peroxide to water. So to keep glutathione peroxidase in activated state, organism has to provide recycling of glutathione. And as we see, recycling of glutathione is provided by NADPH molecules. And the major source of NADPH molecules is pentose phosphate pathway, which is also called hexose monophosphate shunt. In addition to this, we use NADPH molecules for fatty acid synthesis and cholesterol synthesis. And in neural tissue, we use both fatty acids and cholesterol for myelin production. Myelin is extremely important substance for neurons, because from myelin we form so-called myelin sheaths around neuron, which greatly increase the conduction velocity of neural signaling. But most glucose molecules are used in glycolysis. Initially, glucose undergo conversion to pyruvate. Pyruvate enters into mitochondria. Inside the mitochondria, a specific timing-dependent enzyme called pyruvate dehydrogenase converts pyruvate into acetyl-CoA. So, pyruvate dehydrogenase is the second enzyme that cannot function without timing. Acetyl-CoA withdrew into the Krebs cycle, which is also called tricarboxylic acid cycle. and inside the Krebs cycle, acetyl-CoA undergo conversion to alpha-ketoglutarate. Alpha-ketoglutarate is not very interesting substance, because it's just an intermediate metabolite in the Krebs cycle. But what is interesting is that to convert alpha-ketoglutarate to succinyl-CoA, we need alpha-ketoglutarate dehydrogenase, which is thymine-dependent enzyme. So alpha-ketoglutarate dehydrogenase is the third enzyme that cannot function without thymine. Succinyl-CoA then goes through the Krebs cycle, and Krebs cycle results in production of energy in form of ATP molecules. So we require thymine for glycolysis, and as we know, glycolysis is a major source of energy. In addition to this, we have to know that to convert branched-chain amino acids to acetyl-CoA, we require alpha-ketoacid dehydrogenase complex, and the cofactor of this complex is thymine. So it's the fourth enzyme that cannot function without thymine. So what happens with thymine deficiency? With thymine deficiency, transketolase stops working. Without transketolase, we cannot produce ribose 5-phosphate, so nucleotide synthesis will decrease. Also, without transketolase, shunt mechanism becomes disrupted, because without transketolase, the production of fructose 6-phosphate and glyceraldehyde 3-phosphate decrease, and it's the intermediate metabolites of gluconeogenesis. With decrease in formation of new glucose molecules, the income of glucose to pentose phosphate pathway decrease. As a result, the production of NADPH decrease. And because pentose phosphate pathway is the major source of NADPH molecules in our organism, disruption of pentose phosphate pathway causes substantial decrease in NADPH production, and this will have dramatic consequences. Without transketolase, first of all, the production of ribose 5-phosphate decrease, which will cause decrease in nucleotide synthesis. And recall that we need nucleotides for production of DNA and RNA, which are the essential molecules for replication and regeneration. So, with decrease in nucleotide synthesis, cellular division decrease and thereby regeneration decrease. Without transketolase, the production of NADPH decrease. First of all, without NADPH, we cannot provide the reduction of glutathione, and without reduction, glutathione remains inactive. And because glutathione plays a crucial role in antioxidant defense, if glutathione remains in inactive form, the risk of oxidative injury greatly increases. Also, with decrease in NADPH, the synthesis of fatty acids and cholesterol decrease. Without fatty acids and cholesterol, we cannot build myelin sheaths, and without myelin sheaths, the conduction velocity of neurons decrease and areas of demyelination begin to appear. Another pathway that becomes disrupted with thymine deficiency is glycolysis. First of all, with thymine deficiency, pyruvate dehydrogenase is not working properly. And without pyruvate dehydrogenase, we cannot convert pyruvate to acetyl-CoA. So the amount of acetyl-CoA will decrease. And because acetyl-CoA is the major substrate for Krebs cycle, with decrease in acetyl-CoA, the activity of Krebs cycle decrease, and thereby the amount of alpha-ketoglutarate decrease. But in addition to this, with thymine deficiency, alpha-ketoglutarate dehydrogenase becomes dysfunctional. First of all, this causes buildup of alpha-ketoglutarate inside the mitochondria, and also this causes decrease in production of succinyl-CoA, and thereby this causes decrease in activity of entire Krebs cycle. And without Krebs cycle, energy production decrease. So thymine deficiency causes mitochondrial dysfunction that in turn creates energy deficient state. But accumulation of alpha-ketoglutarate can also become very dangerous. The reason is that alpha-ketoglutarate with alanine form glutamate, and glutamate can markedly increase the production of reactive oxygen species inside the mitochondria, which also leads to mitochondrial dysfunction. And with time, constant production of reactive oxygen species can cause substantial neuronal damage. In addition to this, with thiamine deficiency, alpha-keto acid dehydrogenase complex is not working. This causes accumulation of brain-chain amino acids inside the mitochondria that with time can also cause mitochondrial dysfunction. So, thiamine deficiency disrupts two very important metabolic pathways of glucose. It's pentose phosphate pathway and aerobic glycolysis. So, the question is, how disrupted biochemical pathways cause tissue injury? In case of Wernicke-Korsakoff syndrome, we are interested in brain tissue. So, thiamine deficiency causes mitochondrial dysfunction, energy deficiency also it creates pro-oxidative state, and altogether this causes severe injury to brain cells. And injury to brain cells eventually leads to cytotoxic and vasogenic edema. To explain this, we have to know the structure of blood-brain barrier. So here we have blood compartment and brain compartment. In the blood we have a lot of fluid, and brain tissue is separated from the blood by blood-brain barrier. The major components of the blood-brain barrier are endothelial cells, basement membrane, and astrocytes. Also, endothelial cells are connected with each other by tight junctions. So, in normal condition, blood-brain barrier has low permeability. And once some little amount of fluid enters into the brain tissue, it's very important for neurons in the brain tissue not to let all this fluid to enter inside them. So, neurons have to maintain their normal permeability. And neuronal permeability is maintained by pumps. First of all, it's sodium-potassium ATPase, and important that this pump cannot function without ATP, so this pump is energy dependent. The function of this pump is to pump out sodium in exchange for potassium. So this pump creates low intracellular sodium level, and the reason why it's extremely important is that low intracellular sodium permits to maintain normal membrane potential, and membrane potential permits to drive chloride out of the cell. As a result, we have sodium and chloride outside the cell and potassium inside the cell, and this ion balance determines cellular permeability. So let's suppose that we have 3 neurons and they all function in a similar way. So they require a lot of ATP to maintain this balance between sodium and chloride outside the cell and potassium inside the cell. But sometimes something goes wrong. The first possibility is cytotoxic edema, in which disruption of cellular metabolism causes cell swelling. We know that thiamine deficiency disrupts aerobic glycolysis, and because glycolysis is the major source of energy, this immediately creates energy deficiency. And in energy deficient state, energy-dependent pumps as sodium-potassium ATPase are not working. Because pumps are not working, this causes accumulation of sodium inside the cell. Increasing intracellular sodium causes rapid depolarization. And with depolarization, chloride not only not leave in cell, but instead chloride begin to enter into the cell. And because sodium and chloride are osmotically active, they attract water. So accumulation of intracellular sodium and chloride force fluid to enter into the neuron, and increasing intracellular fluid results in cell swelling. Exactly this state we call cytotoxic edema, and it's very dangerous complication because if cytotoxic edema lasts for a prolonged period of time, it will cause apoptosis. But also we have vasogenic edema, where the major problem is abrupt increase in permeability of the blood-brain barrier. As we said, thiamine deficiency creates pro-oxidative state, because glutathione remains inactive, and also there is increase in reactive oxygen species production due to the accumulation of glutamate. And glutamate can markedly increase the production of reactive oxygen species inside the mitochondria, which can cause severe oxidative injury. Oxidative injury can disrupt tight junctions. In addition to this, glutamate itself is highly osmotically active substance. Glutamate tends to accumulate inside the astrocytes, and glutamate is highly osmotically active. So glutamate will force fluid to come into astrocyte by osmotic gradient. Increasing intracellular fluid inside the astrocyte causes astrocyte edema. And because astrocytes are one of the key components of the blood-brain barrier, swelling of astrocytes can greatly increase permeability of the blood-brain barrier. So now more fluid will enter into the brain tissue. In addition to this, damage to tight junctions can also cause increase in barrier permeability. So now this excessive fluid will enter into the brain tissue, where it will cause brain tissue edema. Exactly this state we call vasogenic edema. On this picture we can see the most common sites of brain injury in case of Wernicke syndrome. And probably mammillary bodies are the most vulnerable anatomical structures. So how we can diagnose brain injury in case of Wernicke syndrome? Usually, cytotoxic and vasogenic edema cause hyperintensity on MRI images. So here we can see hyperintensive areas in different anatomical regions of the brain. Some of them are more intense, some are less intense, but hyperintensity should be present. And on this image, we can compare normal image with image of patient with Wernicke-Korsakoff syndrome. And the biggest difference is the shrunken mammillary bodies in case of Wernicke-Korsakoff syndrome. So mammillary bodies are one of the most vulnerable structures to chronic thiamine deficiency. So how cytotoxic and vasogenic edema manifests clinically? Initially, Wernicke syndrome develops. First of all, encephalopathy develops. Encephalopathy usually manifests with disorientation and drowsiness. The second feature is oculomotor dysfunction, which manifests as horizontal nystagmus and bilateral nucleus palsy. And the last feature of Wernicke triad is ataxia. Usually it's postural ataxia or gait ataxia. But with time, chronic damage to brain tissue can cause not only temporal injury, but neuronal necrosis, which is an irreversible condition that is characterized by death of entire areas of brain neurons. And now, such severe condition cause formation of new symptoms, which we know as Korsakoff syndrome. First of all, it's memory loss. It can be anterograde or retrograde. The second symptom is confabulation, and the third feature is personality changes. So, Wernicke syndrome is an acute condition caused by damage to neurons, but with proper treatment this condition can be reversed. So, the prognosis is not bad. But Korsakoff syndrome is a chronic condition that is caused by neuronal necrosis, and because of this it's practically irreversible. So the prognosis is very bad. The easy way to memorize ocular dysfunction in Wernicke syndrome is W, because we can add eyes to these W peaks, and this will tell us that W is associated with ocular abnormalities. And the last question is how to treat. The treatment is very simple. Initially, we give thiamine. With thiamine, pentose phosphate pathway begin to work. First of all, nucleotide synthesis increases. Also, antioxidant defense improves, and myelin synthesis increases. Also, with infusion of thiamine, enzymes involved in glycolysis begin to work once again. So we restore energy production and reverse mitochondrial dysfunction. And in addition to this, thiamine reactivates the utilization of brain-chain amino acids, which also reverse mitochondrial dysfunction. And after thiamine, we usually give glucose, because now the pathways are working, So if we give glucose, this will accelerate energy production, nucleotide synthesis, etc. But the most popular question about Wernicke syndrome and the most common mistake in clinical practice is to give glucose first and then thiamine, or just to give glucose. The problem with glucose is that because pathways are not working, we cannot utilize glucose in pentose phosphate pathway. So additional glucose will give us additional pyruvate molecules. But we cannot convert pyruvate into acetyl-CoA because pyruvate dehydrogenase without thiamine is not working. So glucose infusion will give us massive amount of additional pyruvate. And the problem is that damage to neurons will create acidic environment. And acidic pH force conversion of pyruvate into lactate. And accumulation of lactate inside the cell will cause severe lactic acidosis. And even if some thiamine reserves are still present, and mitochondria will be able to convert some amount of pyruvate into alpha-ketoglutarate, we cannot convert alpha-ketoglutarate to succinyl-CoA, because alpha-ketoglutarate dehydrogenase is not working. So this will cause just additional accumulation of alpha-ketoglutarate and thereby glutamate inside the mitochondria. And increase in glutamate will induce additional formation of reactive oxygen species that will cause just additional neuronal damage. So infusion of glucose before thiamine can only worsen initial condition. It's probably the most popular question related to Wernicke-Korsakoff syndrome.